Good morning, everybody. Welcome to the new cohorts of students in the Department of Entomology and Plant Pathology. My name is Marcin Nowicki. I am a research assistant professor with the department, and I am very happy to show you some snippets of the activities in my lab. Let's get started. What I chose to show you as the introductory slide is perhaps the gist of the information that I'm showing in the first paper that I published from my work with the EPP in 2018. This paper, published in PLOS ONE, analyzed the haplotypes of the cultivars of ornamental dogwoods, Cornus florida, and the Asian counterpart, Cornus cusa. You're seeing here the comparative genomics analysis of the chloroplast DNA, and for more details, you are cordially invited to read the paper. As scientists, we are blessed to be able to travel the world for professional purposes, and I definitely took advantage of that. I started my adventure with sciences working for my master's in Warsaw University of Life Sciences, where I analyzed plant nitrogen biochemistry and assimilation. Later, I moved to Hiroshima University in Japan for a one-year exchange program on molecular biology. Then I continued my studies for biochemistry in plant lipids department in Aachen, western part of Germany. Later on, I moved to a short postdoc with Max Planck Institute to analyze techniques for plant cell wall biochemistry. And then with the whole group, we moved across the ocean to Michigan State to continue this postdoc appointment for a couple more years. After this, I went back home and worked for 6 years in a Research Institute of Horticulture in my hometown, focusing on the vegetable plants molecular breeding. And in 2016, I joined the laboratory of Dr. Robert Trujillo in EPP to work on molecular population genetics and on invasive species. In this capacity, I was also promoted last year to the ranks of research assistant professor as a PI of my own lab. And even though I learned English for the better part of my life, I still retained a little bit of my native accent. So this is kind of distinctive characteristics of mine. Asian connection runs deep because I went to Japan, as I introduced you earlier, but I also visited Beijing 2 years ago to attend a conference on horticulture research. For the same conference, I also was fortunate to go to Italy last year. And it is, of course, important to keep hobbies and to keep your mind off the overload that can happen easily. So for those purposes, friends are very helpful, and multiple activities are very helpful as well. Here, for instance, I'm showing you that in Knoxville, I made great friends with whom I was playing euchre, and this single hand enabled me going solo to finish the game because it earned us a extra point. I love Star Wars and I love board games. So this is also a pastime activity of mine when research and thinking goes too intense. Not only human friends are important, but also the pets are important as well. And Knoxville is, as you probably know by now, a very dog and pet friendly city, which is another fortunate happening for me. In EPP in particular, it pays off to make connections. It pays off to make great friends because with this fun crowd, we managed to do all sorts of activities, from going out to enjoy the hockey game to actually establishing our own volleyball league Tuesdays and Thursday afternoon before COVID struck. We're also in this great place that we have Oak Ridge Institute, a very short drive from us, and I strongly encourage you to take advantage of this fact. So when in the lab, of course we work, but you know, a little fun on the way never hurts. So you can see me sampling the pistachios right here in the middle picture, but I'm also working hard de-icing the freezers, which is on a daily nuisance, if you will. Um, let's go to science. My background here is in population genetics, and this is a very powerful approach to analyze the diversity of the species. So in one of the projects that I'm writing up right now, we analyze the downy mildew pathogens, which are obligate oomycete pathogens. And you can see that here we show the diversity and the distinction that the population structure approach, population genetics approach, and allowed us to notice between downy mildews of cucumbers, Pseudoperonospora cubensis, and these of Pseudoperonospora humuli, which infects hops. Even more so, we noticed that the multiplication mode of the pathogens differ and distinguish the two. So cubensis, or the cucurbits pathogen, is mostly clonal in character, which is signified by the difference from zero value, whereas the humuli pathogen, the hops pathogen, is still multiplying sexually, which of course bears implications for the pathogen management. When looked into more detail, and when we looked into spatial structure, we separated the historical samples from the current samples. And for cubensis, we additionally separated the European samples from the American samples. So we see that the historical samples of the cubensis actually were also multiplying sexually, but likely fungicide control of the pathogens made them evolve, and as such, human activities actually presented selection pressure on the pathogen. About the invasive species, my current project focuses on the Colerepair, which you can see everywhere, and I mean everywhere, come February and early March. This is the very first species to blossom on this winter bear panorama, so I'm sure you will see it around. My student Shivani, who is working with Colerepair, was working very hard to present her first poster on the conference, on the national meeting in her second semester, and it was huge success. And right now we have the paper on this story finalized and circulating among the authors, whereas Shivani herself is focusing on analysis towards her second and final project and second and final paper in this study. I also worked with the rubber dandelion and distinguished it from the common dandelion that is considered invasive species across America. Also from the Asteraceae, population genetics analysis allowed us to distinguish a new form, a new subspecies in the Chrysogenum, which is a tiny little ornamental plant. For the phylogeography insights, population genetics certainly helps. Here I'm showing you again about the Cornuscusa story, which actually can be traced back. The origin of the species can actually be traced back to the now Sea of China origin, which looked, the landmass looked completely different 20 million years ago when the species actually diverged. So this brings me back to my first piece of advice for you, which is use every opportunity you can grasp. And this is because, as I'm showing you here, my PI from Japan was actually the guide for the USDA expedition that sampled the Kusa that I used 20 years later in my research. So this story, the Kusa story that was researched by Logan, our undergrad, won him the Eureka across the campus, across the whole UT, and we were very pleased that this story was shared this year. A few final words of my so-called wisdom. Do your research. This is the time for you to read as much as you can possibly, can, because this will help you in your lab research, and this will show you how other people run or solve a given problem. for your research in the lab, you certainly need to focus on that as well. But again, your background knowledge or your theoretical knowledge that you can learn from the papers will essentially help you go faster in the lab by avoiding somebody else's mistakes. And finally, please remember that you are in EPP. And this is perhaps the most helpful of the places that I was privileged to work at, with all students being super helpful and staff and faculties as well being here for you. So I would strongly encourage you to take advantage of this fact as well. And if you ever run into problems, just give us a shout, and I'm sure we'll all very happy to help you. With that all said, I am also inviting you to my lab webpage for some news and additional insights. And whenever you see me on the corridor, please make sure to say hi and introduce yourselves. Thank you very much for your attention, and I hope for some questions.